Hello everyone. Today I'm going to talk about my research, which focus on investigating the role of RIP4 in melanoma, especially in the context of inflammation. Oh, something is broke. Okay, right. So, melanoma is considered the most dangerous type of skin cancer developed from melanocytes, which are the cells responsible for synthesis melanin. And this is the pigments that protect us from damage caused by solar ultraviolet radiation. Dramatic increase in melanoma diagnosis has been observed over the last 3 decades, especially in young adults. So if melanoma is diagnosed at an early stage, it can be successfully treated just with surgery. But the main problem is that if melanoma is diagnosed at an advanced stages, then are extremely resistant to conventional therapy and even more advanced therapies such as targeted therapies or immunotherapies. And activation of nuclear factor kappa B is believed to be promote melanoma progression. This is because it can regulate the expression of hundreds of genes that regulate important physiological processes such as inflammation, proliferation, and regulation of cell division and cell death. In melanoma, a number of NF-kappa B regulated chemokines are constitutively expressed at high levels. For example, interleukin 8 or MCP1. And it is known that overexpression of interleukin 8 in primary melanoma leads to metastatic tumor growth. So it's not good then. Okay. So I have some problem. And recent work suggests that RIP4 activates the NF-kappa B pathway, but the exact mechanism is still unclear. And early RIP4 study focused mainly on keratinocyte differentiation and proliferation. And now it's more attention is focused on the role of RIP4 in the cancer progression. There is a strong evidence that this kinases is involved in the regulation of K-signaling pathways in a cell, for example, like I say, NF-kappa B, but also WNT, or can act as a linker between different signaling pathways, for example, protein kinase C and NF-kappa B. Recent work suggests that RIP4 may perform oncogenic function, for example, in pancreatic cancer, but what is more surprise, there is also a strong evidence that it can act as a suppressor in a liver or a lung cancer. So it can perform dual function. Okay, so recently we published our results showing that the expression of RIP4 is definitely higher in melanoma cells than in normal melanocytes, both at the mRNA level, so you can see this on panel B, and panel A, sorry, and at the protein level, so this is the panel B. We investigated the expression of RIP4 in established melanoma cells line. So you can see the Western blot results on the right side of panel B. And also we check the expression of RIP4 as in patient-derived melanoma cells line. So this is DMBC cells line, and NHAM. This is the normal human melanoma, normal human epidermal melanocytes. Also, in both cases, we used PANC1 cells. This is a positive control because this is pancreatic cancer cells line. So we know that they have expression of RIP4. In the melanocytes, we don't detect any RIP4 in this cells. So next, to assess the importance of RIP4 in melanoma development, we perform immunohistochemical staining on primary and metastatic melanoma tissue, including samples from the same patients. And we found that RIP4 is heterogeneously expressed in a clinical material. Actually, we see no significant difference between primary and metastatic melanoma. Thus, in both cases, RIP4 was staining from low. So you can see this on the left side. And also, we detect a very strong intensity, what means it's a strong express RIP4 in this samples. So you can see this on the right side. Interestingly, we observed two different types of staining pattern in the cytoplasm. It's a granular and a granular. So when you see the picture of representative image of staining, and in the right side there is high RIP4 intensity, and you can see the difference in the staining pattern. So here you can see agranular staining, and here is a granular staining pattern. So next, to direct address the role of RIP4 in melanoma progression, we examined the effect of ectopic RIP4 downregulation of on two melanoma cells line by using two different RIP4 specific RNA, and we were able to decrease RIP4 levels by 80 to 90%, so with good efficiency. RIP4 downregulation decreased phosphorylation at AKK alpha beta complex and at P65 in both cells line. This means that signal transduction along this pathway is partially inhibited. This is because phosphorylation of AKK alpha beta leads to phosphorylation of A kappa B inhibitor protein, and when it's phosphorylated, then is direct for degradation proteasome, and then P65 and P50 are released and can translocate the nucleus and regulate the expression of hundreds of genes. Okay, so we see that a signal pathway in both cells line was decreased after RIP4 silencing. So next we wanna check what's happened with interleukin-8 in this situation, and inhibition of signal transduction has effect on expression of interleukin-8. So we demonstrated this in an experiment in which cells with decreased RIP4 expression were stimulated by TNF. So as you can see, downregulation of RIP4 kinase leads to lower interleukin expression after TNF stimulation, of course, compared to the negative control. So, in conclusion, expression of RIP4 is higher in melanoma than in normal melanocytes. Downregulation of RIP4 decreased the level of phospho AKK alpha beta and phospho P65 in melanoma cells, and the regulation of interleukin 8 expression by TNF alpha in the melanoma cells was mediated through RIP4 and NF-kappa B activation. So if you need more information, there is our publication, our paper where we more describe this process. So thank you for your attention.